My name is Paul Diehl. I'm the Chief Operating Officer of Cellecta Incorporated, a company located just south of San Francisco, Mountain View, California. We are a company focused on providing research products and services for investigators looking at gene function. Approximately 12 years ago, the entire human genome was sequenced, and that provided the sequence information and sort of the number of genes that are there. But still, most of the genes are not really well characterized, and researchers don't understand how they're controlling and regulating various biological processes and disease progression. So over the last few years, next-generation sequencing has become much more accessible and much more economical, and also oligosynthesis and development of oligonucleotides and gene synthesis. And so we try to make use of these newer technologies to develop assays where multiple parameters can be looked at the same time, and then using next generation sequencing and barcoding and things of that sort the individual components of the system can be sorted out to find out really what are the essential elements in the process. We've actually developed two different platforms at the moment that are using this sort of approaches, and one is really focused on doing large-scale pooled screening using loss of gene function across the genome, and we've done this using the CRISPR or RNAi technology. And so we offer libraries and screening services so that researchers can look at genome-wide or targeted sets of genes and understand how the gene disruption affects the various phenotypes that they're looking at, in particular often viability of the cells. And so they're often looking for essential genes. Like, for example, if you're looking in cancer cell lines, you want to understand which genes are essential for viability of those tumor cell lines and see how you could kill cells. And another approach we've taken is to try and make full-scale transcriptome analysis or gene expression analysis more accessible. And we've done that by trying to combine the advantages of RT-PCR, where you can amplify total RNA, with the, again, next-generation sequencing technology. And so we have an approach where we have targeted amplification of transcripts across the genome, genome-wide, that then can be sequenced, and then based on the sequencing information you can look and see which genes are up or down regulated in response to various biological processes. Well, we are a technology-focused company, and we're continuing to develop new approaches, and we want to continue to sort of push the limits and make the technology more accessible and enable research scientists to do this sort of gene function analysis. Because the more researchers that are working on these type of projects, the more information that's out there, and then the faster discoveries are coming for various diseases, and our understanding of biology is advancing.